All right, everyone. Thank you very much for coming along to my talk about mulloway movement in the Great Australian Bight. We'll get right into it because there's quite a bit to get through today. So we're going to talk about mulloway movement in the eastern Great Australian Bight and relate that information to the marine parks in the area. We'll discuss some problems we have with the novel sort of tracking approach that we used, and also hopefully I'll have time to talk about some broader themes associated with large demersal fish movements. So a little bit of background about the study species. Mulloway are from the family Cyanidae. Large body cyanids are generally big, well, they're large silver fish with big silver flanks. They have developed otoliths, large otoliths for their body size, and swim bladders, which they use to communicate, possibly in turbid conditions, turbid waters. They're good eating. There's fisheries worldwide fishing cyanids, and yeah, mulloway is a typical sort of example of that. Let's go back. The fish in the lower left-hand corner is a mulloway. That's a red drum, another cyanid, very similar looking animals. That's sort of typical for the family. They're widely distributed in Australia. Indeed, they're the only southern Australian cyanid mulloway, and they do exhibit population structuring despite their wide distribution. In South Australia, we have two populations. One centred in the Gab, the focus of today's talk. The other one is sort of centred around the Coorong and the southeast of South Australia. Yes. I mentioned they're fished and they're good eating, they're fun to catch. There's enormous pressure, or there's significant pressure from the recreational sector in the eastern Gab. These magazine clips sort of highlight that. These fish sell magazines. It's extremely popular recreational fishery. There is also commercial fishing in the area, but what's quite unique about this area is the recreational pressure is substantial. Yeah, if you want to, we did try and quantify that recreational pressure, which is something that's traditionally hard to do. For more on that, you can check out our report. Okay, so the mulloway population at Yellowton, it's isolated, it's fished, it's under pressure, enter the marine parks. These provide a possible way of protecting mulloway, slowing the harvest, keeping things sustainable. I will point out that today's talk I'll sort of focus mainly on the state-based marine parks and maybe just touch on some of the Commonwealth marine park stuff as well. So one super cool thing about our mulloway research in the Gab is the local community involvement. Actually, the local Indigenous community were one of the first, or the first people to raise concern that the mulloway harvest was not sustainable in the area. And the community has been very hands-on in field work, funding, and all sorts of facets of the project. So, yeah, that is definitely worth noting. It was a pretty nice office down at Yellowton Beach as well. Here we have Dr. Paul Rogers on the right there, talking about the ins and outs of satellite tagging. He's an expert in that field, and yours truly talking about some biological aspects of mulloway. Yeah, it's a really nice place to go. So just to summarise that extensive background information, isolated and vulnerable population at Yellowton, unique gene pool. For more on the genetics, the population genetics, you can check out my publication. In response to the Indigenous community and local communities' concerns about the harvest of mulloway being unsustainable, we formed a research group. We've looked at various aspects of the mulloway population biology there, and including the movement around the new marine parks, which today's talk is focused on. So just to be clear on the objectives, we want to determine what sort of protection the parks provide mulloway, given that they're quite heavily exploited by the recreational and commercial sector, and in doing so test a novel approach to tracking large marine demersal fish. Excuse me. Okay, so to track demersal fish and the satellite tags, we needed something that was fishery independent. We didn't want to rely on recaptures to get our movement information. Also, it's not feasible in such an isolated area to use receivers and acoustic methods. So the satellite tags we thought would be worth a shot. They're almost like a black box recorder of the fish's movement, habitat and environment. Today's focus is on that movement aspect. After a predetermined period, the tag stops logging data and releases from the fish and floats to the surface and starts transmitting a position and also the environmental data, etc. So we thought they were quite a useful method for getting some data on mulloway movement, not tested before on demersal finfish. Excuse my spelling of finfish there. And we had to do quite a lot of R&D on the method because there's nothing in the literature about attaching them to large finfish. Once we developed an anchoring system to tether the device to the fish, we got down to Yellowta Beach and started trying to catch mulloway. Once again, back to that good office of Yellowta Beach. We angled them with heavy surf tackle, trying to get them in as quickly as possible. Got them in as quickly as possible, performed surgery on the beach, inserting a dart which tethered the tag to the animal. Then we released them as quickly as possible. Also, whilst in the area, we collected some genetic material and other tidbits to do some complementary studies, as I mentioned, the population genetics at the same time. So, the results of our tagging effort, we tagged 19 fish over various summers and autumns over a 3-year period. Fish were sort of medium to large. 11 tags reported to the Argos satellite network after nominal periods of 100 days. 8 tags were never heard from again. That meant we had a success rate of about 58%, which is comparable to other studies, but it was disappointing that some tags didn't come through expensive. The devices were about $4,000 each at the time. But the ones that did come through provided some amazing data, and particularly on the movement. So, where did they go? What we've got on this map, is just the capture position, which is around Yellowta Beach, and straight lines represent the net displacement or net movement. The black dots and numbers represent where the tags came off. So if we look at number 1, that was the first fish that came through with the goods. That's where we caught the fish. That's where the tag came off. Focusing on these 3 fish, they went west. One individual went over 600 kilometres west from the tagging location. The interesting thing about these 3 individuals is they were all autumn deployments. The 100-day deployment period was all over autumn. So there might be something going on with overwintering. Importantly, at least 2 of them, left the marine parks. Number 4, see if I can get this pointer right, is still just within the marine park network. But yep, so westerly movement in autumn. If we go to the inset, we've got these 5 individuals here that didn't go far at all. The tags came off within the Far West Coast Marine Park. These were all summer deployments. So a little bit of a pattern unfolded where we had a westerly migration or migration during autumn and residency along the beaches in summer. there's anecdotal evidence to suggest the fish spawn there in summer, so that was quite an interesting result. However, there was a couple of outliers. There was 3 outliers. Fish 10 and 11, they were autumn deployments that went east. So they broke the pattern a little bit. Looks like I haven't got time to really go into why. It was a warmer autumn. Also, those individual fish were a bit smaller than normal, so there might be some size-based movement going on. Or fish 3, that was a summer deployment that went east to Cactus Beach. Yep. I think we'll just keep moving, given there's only 2 minutes to go. So what happened between fish capture and pop-up, I should say? The fish, we can sort of rebuild their movements using something called geolocation. Unfortunately for demersal fish, it doesn't appear that crash hot. It relies on timing of sunset and sunrise, and we think that maybe the water column is too cloudy to get accurate representations, because I know that that fish didn't go off the shelf break there, and it all doesn't make sense, so I don't want to dwell on that. We didn't utilise that information, whereas if you were tagging pelagic animals, that would probably be quite useful. Thank you. So what we did get, though, that helped elucidate what happened between capture and tag pop-up is the depth information. The top graph is one individual, the bottom graph another. The fish, this one was one of the westerly migrants, and you can see here at this point here, it goes offshore. I should mention the y-axis is the depth, surface to relatively deep, and the deployment time summarised at the 2-day level. This fish went relatively deep and stayed there. Here's periods in the blue circles where the fish resided in the shallows. The bottom individual was a summer deployment, stayed shallow, except for this sortie out to deeper water, but under 40 metres. In the red circle, we can see where the tags came off the fish and surfaced and started transmitting that data to the satellite system. So just back to this map. The Far West Coast Marine Park has a 40-metre contour along here in yellow. It's hard to see because it actually borders the marine park. So using that information, I could tell that those 5 fish, the summer deployments, were actually residents of the area over summer, but they didn't take off and come back, more than likely anyway. What else? Just to summarise, fish departed the MPAs in autumn and they resided in summer. That sort of fits in with anecdotal evidence or information that the fish are around in summer and not in autumn and winter. So the parks provided partial protection, which is better than nothing, and particularly as they may be spawning whilst in the park system. This type of data is an important step in determining the effectiveness of protected areas. Probably out of time. Yep, so we won't worry about the problems. And the conclusions are probably fairly repetitive, but the take-home message is that they interacted with the parks and appear to come and go. It's probably not feasible to have the parks going into Western Australia, so at least they protect them during spawning, and I might wrap it up there.
Thanks, Tom. Time for one quick question. Can you yell it out, Simon? You've got a big voice.
You say MPA, do you mean sanctuary zone?
Just repeat his question.
Yeah, MPAs have sanctuary zones.
I'm just interested in what the spatial scale you're talking, because
There's various...
are relatively small.
Yes, they are. They are. But at least that's something. And there's other activities are restricted in the other zones as well. Yeah, but there is still potential for capture when they're outside those small sanctuary areas, which I think during November to March, approximately November to April, is just here and here and there. But I don't profess to be an expert in the marine park system. But some protection, yeah.